Hello, welcome to Lesson 5, the structure and function of phospholipids, part of the macromolecules unit. This is the last lesson which will target part B of the assignment. In it we have to look at the phospholipids and we're going to look at the chemical structure and the role in membrane functioning, which we should know a lot about already. Because in our cell biology unit we learned that the cell membrane is made of phospholipids. They have a head and two tails, and you put two of those layers together and you get the phospholipid bilayer, which is the fundamental sort of structure of the cell membrane. And if you remember, some of the properties that we talked about in that unit were that the phospholipid bilayer is waterproof in the main without the channels. It has lots of proteins embedded, lots of sugars embedded, which we've already covered in all the other lessons. It's a very polar molecule, which we'll talk about later. And yeah, it generally protects and keeps as a barrier for the cell contents. And without the cell membrane, we've got no cells, and without no cells, we haven't got any life. So without further ado, lipids are a little bit different to proteins and carbohydrates, because in those two lessons we had a very sort of simple building block that built up bigger and bigger. Well, lipids are a sort of category on their own, and they branch out into many different types of structures. So it's not like you have a small unit and you link them together and you make a big unit. They have similarities, but it's a bit more complex. Now, fortunately for us, we only have to talk about phospholipids in terms of the assignment. So sterols and fats, we won't need to be talking about. We'll keep an eye out on for them, but that's really what we're focusing on in this session, and indeed the unit. Now, if you can see that all of these different functions down the right hand side are all lipid related, and so you can see that they're found, in a variety of different settings within the human body. So I've included a definition of a lipid at the top there. The term lipid has been loosely defined as any of the group of organic compounds that are insoluble in water but soluble in organic solvents. Basically, insoluble means that it doesn't dissolve. So rather like a little bit like sugar in your tea, not a great example, but if you just put sugar in your tea, it dissolves. So that would be soluble. If you put a piece of metal in there, then that would be insoluble, because it doesn't separate, it doesn't dissolve in water. And we'll look at that concept a little bit later on in the slides, hopefully. So that's a bit of an introduction. So this is a fat, all right, not a phospholipid, but I needed to show you this just to show you how the phospholipid molecule is formed. So this is a fat, and a fat is consisted of a glycerol molecule attached to 1, 2, and 3 fatty acids. That's what a fat is. Now, we're not going to go really into detail about fats because we don't need to, but that's where you would look for your saturated or non-saturated fats if you so wish. But what I really wanted to show you is how these fatty acids are joined to the glycerol molecule, and you will not be surprised to see that it's our old friend, a condensation reaction or a dehydration reaction. And you can see that, let's just highlight this in green. We have one fatty acid here that has an OH group on the end, and we also have on the glycerol molecule a hydrogen poking out, and we get the H2O popping off, and we get the condensation reaction. So you can see hopefully that the O is joined here, and so we end up in total with these 3 condensation reactions and 3 molecules of water, and we get a fat. That's how fats are formed. So what the hell has this got to do with phospholipids? Well, hopefully you can see that a phospholipid, if you remember, has a head and two tails. These tails are fatty acids. So let's have a look at those. Here we go. This is a phospholipid. It has the same glycerol molecule sort of head, and then it would have two fatty acid tails. The only difference being that this third fatty acid tail on a fat has been exchanged for this, a phosphate group. Okay, and that's really, really important in terms of the functioning, because the structure of a phospholipid enables it to function of what we've learned. So we've learned that it's a barrier, we've learned that it can attach proteins and carbohydrates and all these kinds of things, but it's the fact that it's different to a fat that enables it to function as a phospholipid and do all those fantastic, wonderful things that we've already learned about. So yeah, the fatty acid chain of a fat is exchanged for this phosphate group over here, and you'll notice that the heads are hydrophilic and the tails are hydrophobic. We know this. These don't like water, these do like water. Why is that? That's the question, and hopefully we can add to your knowledge of what you already know. So when we look at a phospholipid closely, we first of all need to understand what the word polar means. And just like the North and South Pole of the Earth, the head of a phospholipid is exactly the same. So you can see that one end of the phospholipid is positive, one end of the phospholipid is negative, but the two tails are neutral, which means they don't really have a net charge at all. Now this is really, I have to give you a warning, this is really not going to make that much sense if you've missed my atomic structure lessons, where we talk about, you know, we used marshmallows to model different molecules and atoms. If you've missed that lesson, come and see me, let me explain it to you. Now you can carry on listening to this, but I suggest that the next sort of minute or two will not make that much sense to you. Now the question is, why does this have a positive area? Why does this have a negative area? Well, if you charted out all these electrons of all these elements and compounds and molecules, you would find that they're simply that there'll be more protons at this side and there'll be more electrons at this side, whereas this section of the tails are neutral, so they're really happy molecules. And you can see it's a hydrocarbon tail, so each carbon has two hydrogens attached, and it's with a carbon each side, which means they're really happy atoms. You know, they're really happy, and there's no areas of positive and negative, whereas this, you can see, is quite a mismatch of atoms, and therefore it does create a positive end and a negative end. So what does this positive and negative end mean? Well, in order to understand why this phospholipid results in such wonderful features, such as it forming a phospholipid bilayer. So why does it form a phospholipid bilayer? We have to look at also the structure of water. So in this bottom right hand side here, we've got the atomic structure of water, and this is why I said before, if you don't know about atomic structure and electrons, then you're not really going to get this. But see whether you can follow this anyway. Now here we've got a molecule of hydrogen, sorry, an atom of hydrogen, another atom of hydrogen, and one oxygen, which is H2O. H2O. And hopefully you can see that each atom is a happy atom. So this hydrogen here has two electrons on its outer shell, which is happy. This one has two electrons on the outer shell. This one's happy. And if we count them up, 2, 3, 4, 5, 6, 7, 8. The oxygen has 8, which is fantastic. They're all happy. However, you can see that let me try and draw this. On this area here, there is no electrons. But on this area here, you can see that there's 3 electrons. Now I'm only using this as a sort of rough example. But what you can see is that these 2 hydrogens, by pulling each other together almost, they're quite close and they're not sort of opposite ends of the oxygen atom. They actually pull close together by forces which I'm not going to go into, but it just does. You have to accept that. Well, because there are only protons here in the nucleus, there's no electrons on the outer shell, and this becomes a positive area of water. And on the other side of the water molecule, where the oxygen is, this becomes a negative area of water. And so what we can say is that water is a polar molecule. Just like the phospholipid, it's a polar molecule which has a positive end, a negative end of the phosphate head, and a neutral end of the tails. So what function does that have? Well, what we've got here is that polar molecules can like to interact. So when we say that the head, the phosphate head of a phospholipid is hydrophilic, so I'll repeat that because I stuttered myself. We said that the head of a phospholipid is hydrophilic and the tails are hydrophobic. Now the reason why that is because this polar head is charged on a phospholipid. Well, water is also a well, it's not, that's a lie, that's ambiguous. It's got a charged areas, and the same is true of water. So hopefully you can see that it's hydrophilic because it's attracted to water. So the heads are attracted to water. They like to be in water because they're charged. Whereas this neutral section of a phospholipid does not have a charge, and therefore it doesn't like water because that is a charged particle, molecule. And that's what gives us the properties of a sealing molecule. So in the top right hand corner here we've got a micelle, which is called a micelle. So if you just dropped phospholipids into water, it would naturally form this sort of circular unit where the heads, which are polar molecules, like to be with the water, which is also polar molecules. And you can see that they form this sort of this waterproof barrier where there is no water inside. Now that's not the bilayer, but that's just dropping phospholipids in water. You'd get that circular formation of a cell almost, but this is obviously singular formation. That gives the phospholipid bilayer its major properties. And I hope you followed it. I hope I did it justice, but it's not one of the easiest things to sort of explain through video. Now, when we look at the function, if we got a definition of the function of the cell membrane, then we're talking about the definition of cells. They are a barrier. They're more than a barrier. It controls what passes in and out of cells. It provides structure and allows cells to communicate with it. Well, we've already said about proteins and carbohydrates, but it's the proteins and carbohydrates that are sat in the phospholipid bilayer, so they have a very much major role in what proteins and carbohydrates do. So actually a phospholipid bilayer is a solvent, which means things are sort of dissolved in it, attached to it. So that's one of the reasons why carbohydrates can attach or dissolve in phospholipids, because they can communicate, and same with protein, should I say. Now, in terms of what the main area I want to talk about in this slide is that it's a barrier. We know that. We know the cell membrane is a barrier. And actually, the cell membrane is a self-healing unit, which means that if you had a cell membrane here and you sort of took a chunk out of it, will my eraser work? No, it will not. If you have a cell membrane here and you took a chunk out of it, let's say, then the phospholipids would be attracted to each other and they would seal up. It's a self-sealing molecule, which what I'm trying to get at is you've got to explain the structure and function of a phospholipid bilayer, basically. And what I'm saying is that the reason why phospholipids is brilliant for a cell membrane is because it's self-healing. And the reason why it's self-healing is because of the structure of the phospholipid, in that it's charged and there's a neutral area, and hydrophilic heads and hydrophobic tails, and that's the reason why it's self-healing. It's also flexible, so lipids at the right temperature are really flexible, really fluid, which helps it move and protect from the environment. But we also get this, that should say flip-flop, not fit-lop. What we get if we draw the phospholipids here, the flip-flop, actually this phospholipid can flip inside and this phospholipid can flip outside. So if this had a sort of a glucose chain on it, this could flip on the outside, and that's what you get. I think I read on this article down the bottom that it does so about once a month, but it might do it more than that if I read into it. And the phospholipids are able to exchange. So if we draw a phospholipid here on the bilayer, and we drew a phospholipid here next door on the bilayer, then actually this phospholipid could swap with this phospholipid. So they're really, really flexible, really, really fluid. They're able to self-seal, they're able to attach sort of proteins in it, so they're able to be solvents. And that's really what you need to try and get across in your assignment, with obviously sources and showing that you've read around the subject. Hopefully that gives you the sort of skeleton answer with quite a few trimmings on to try and explain the chemical structure of phospholipids, which should be really, really straightforward. There's a real quite difficulty here in showing whether you understand the structure of a phospholipid and how it relates to function. Now you're asked to describe its role in membrane functioning, so you're not really expected to show a really, really, really thorough understanding of how that structure of a phospholipid relates to its function, but I would very much like you to have a go. Show as much understanding as you can, but do not panic, do not worry if it's not fully sinking in, particularly if you've not done the atomic structure lesson. But I hope this gives you a good fundamental start to trying to explain the chemical structure of phospholipids and how they play a role in membrane functioning.